AHL Topic 8 IB Biology, subsection 8.2 Cellular Respiration. Alrighty, so before we look at topic 8, let's revise what we covered in subsection 2.8, which are our foundations for this unit. So we looked at cellular respiration. We said it's a controlled release of energy in the form of ATP. We said it could be aerobic or anaerobic, not of anaerobic. Aerobic respiration involves 3 stages. So in 2.8, you just needed to know those 3 stages. And in topic 8, you need to know them in much more depth, and we even add in an extra stage for you. You knew that respiration involved organic compounds reacting with oxygen to produce carbon dioxide, water, and energy. We discussed that food is the fuel for cellular respiration, specifically carbohydrates and sometimes some fats, as that is now telling you. We also discussed that cellular respiration was a catabolic pathway, meaning it releases energy by breaking down complex molecules. Sounds quite simple, breaking down complex molecules. We'll see if you think it's simple by the end of the video. Okay, so this is an overview of cellular respiration, and at 2.8, this is where you need to stop. However, in topic 8, we're now going to look at glycolysis. We're going to add in the lovely link reaction. We're also going to look at the Krebs cycle and the electron transport system. Before we get there, let's have a think about this product called ATP, this substance ATP, adenosine triphosphate. Yet again, making a video so I cannot speak. Okay, this is a chemical that can diffuse to any part of the cell and release energy. It's made from organic compounds in the cells through cellular respiration. So that is from topic 2.8. Okay, so ATP plus H2O gives you ADP plus a phosphate plus energy. ATP is hydrolyzed to form ADP. That's inorganic phosphate and energy. This is what we call an exergonic reaction, or exergonic. The inorganic phosphate will attach to another ATP molecule. The new ATP molecule is then phosphorylated. So we get ADP plus our free phosphate, then moves on to ATP plus water. Now, if you're interested in what an exergonic reaction is, that is a chemical reaction where the change in free energy is negative. Okay, whereas an endergonic reaction is obviously the opposite, where the change in free energy is then positive. So exergonic, negative change in energy, endergonic, positive change in energy. So, cell respiration involves the oxidation and reduction of electron carriers. So that's one of your understandings. What does this mean? In the equation, glucose is oxidized because electrons are transferred from it to oxygen. The protons found in the electrons are then used to produce water. The oxygen atoms that occur in the oxygen molecules on the reactant side of the equation are then reduced. Because of this reaction, there is a large drop in the potential energy of the compounds in the product side of the equation. Redox reactions can play a key role here in the flow of energy through living systems. This is because the electrons that are flowing from one molecule to the next are carrying energy with them. In a similar sort of way, the catabolic and anabolic pathways mentioned earlier are also closely related to one another. And you can see that in our equation there, just not in quite so much depth. All right, so what electron carriers are we talking about? We're talking about our NAD plus and our NADH. Okay, these are substances that can accept and give up electrons as required. For this reason, electron carriers often link oxidation and reduction in cells. The most common carrier in respiration is NAD plus. Hydrogen atoms have one proton and one electron. When two hydrogen atoms are removed from a substrate when two hydrogen atoms are removed from a substrate, NAD plus accepts the electrons from both atoms and a proton from just one of them. Okay, so we've got our NAD plus, we've got our two hydrogens, and basically that's what we then get. That NAD plus is accepting the electrons and proton from one of them, so that's why we get NADH, and then we get a H plus because an electron's been taken from that hydrogen. Okay, so we've got our NAD plus our two hydrogens, which is giving us NADH, which has no charge because it took the electron from the free hydrogen to neutralize the original NAD plus. And we've got that hydrogen there. It's got a positive charge because its electron has been attached to the NADH. That's just showing what we just discussed. Alrighty, let's then have a look at our first process. Our first metabolic pathway in respiration is glycolysis. So there we are, back to our overview, looking at glycolysis. So just a general overview of glycolysis. It's the splitting of the sugar, the first step of cellular respiration. It's the breakdown of glucose molecules into two pyruvate molecules. One 6-carbon sugar is broken down into two 3-carbon sugars, which are the pyruvate. It takes place in the cytosol of the cell. Oxygen is not required for glycolysis. And here we have glycolysis, a linear metabolic pathway. You do not start with, your product is not your initial reactant, so it is linear. Alrighty, so part of glycolysis is what we call phosphorylation. It is the addition of a phosphate molecule to an organic molecule. The purpose is to make the phosphorylated molecule more stable and more likely to react. Okay, so glycolysis, step 1, phosphorylation. Glucose is converted to glucose 6-phosphate. And if you look in that diagram there, you can see what that means. This is coupled with the hydrolysis of two ATP molecules, which donate the phosphate groups. The ATP is converted to ADP as the phosphates are attached, so that's as we previously looked at. Then there is a release of energy that is required for the next step to occur. Because the reactions are coupled, the combined reaction proceeds spontaneously, so it just happens. Okay, so after phosphorylation, we then have step 2, which is lysis. And this is the most significant consequence of glycolysis in the production of a small yield of ATP without the use of oxygen by converting sugar to the pyruvate. Okay, so we've got our sugar molecule and we're breaking it into the 2 pyruvates. This cannot be done in a single step, but instead requires many small steps in the form of a metabolic pathway. So first of all, fructose 1 and 6 biphosphates is split into 2 triose phosphate molecules. Sorry, the fructose biphosphate is split into 2 triose phosphate molecules. Lysis is the part of the reaction which involves the active splitting of the substance and is partly where the name glycolysis comes from. Fructose 1,6 biphosphate is split into 2 equal molecules called triose phosphate. I said that. That was said up there. This is a repeat. All right, glycolysis step 3. So we've got our 2 pyruvate molecules. What's happening now? Triose phosphate is oxidized, loses a hydrogen, which provides enough energy for it to gain a second phosphate and becomes 2 molecules of glycerate 3 phosphate. Energy is produced by removing 2 hydrogen atoms from each triose phosphate molecule. This energy is used to add another phosphate group to each of the triophosphates, which means we now have a compound with 3 carbons and 2 phosphate groups. So just look at your diagram there. An oxidation reaction, which means the loss of hydrogen from triose phosphate. The 2 hydrogen atoms removed are accepted by a hydrogen carrier named NAD+, which we've already looked at, which becomes NADH plus our hydrogen ion, which is also known as reduced NAD, a reduction reaction. The gaining of hydrogen by NAD+. Step 4 of glycolysis, ATP formation. So we've got our 2 pyruvate molecules are formed as 4 ADP are phosphorylated, forming 4 ATP molecules. The final product, pyruvate, is formed by the previous 3 carbon compounds losing 2 phosphate groups. The phosphate groups are lost from the carbon compound and are added to the ADP molecules. As we have 2 phosphates attached to each compound, we have 4 spare phosphates overall. This means we can convert 4 ADP into 4 ATP. In other words, we produce 4 molecules of ATP. However, because stage 1, the phosphorylation, uses 2 molecules of ATP, overall we are producing 2 molecules of ATP. We do, at the end of these videos, look at the ATP balance and ADP balance for all the steps of respiration. So a summary of glycolysis. Glycolysis occurs in the cytoplasm of the cell. There are 4 main parts in glycolysis, not including intermediary steps. First of all, we have phosphorylation, followed by lysis, followed by oxidation, followed by our ATP formation. Overall, one molecule of glucose results in 2 pyruvates and 2 NADH plus hydrogen ions and 2 ATP. That is our net gain. Moving on now to the next stage of respiration, which is called the link reaction. This is where our pyruvate becomes acetyl coenzyme A, or is transferred to acetyl coenzyme A. So what happens to our pyruvate? Glycolysis has taken place and 2 molecules of pyruvate have been produced from every 1 molecule of glucose. If oxygen is present, this pyruvate is absorbed into the mitochondria, where it is then fully oxidized. So the link reaction, this is where our pyruvate gets oxidized. Carbon dioxide is removed from the pyruvate. This is the decarboxylation, i.e. the removal of carbon. Pyruvate is oxidized by the removal of hydrogen. The hydrogen atoms are removed by NAD+ to form NADH plus hydrogen. Can you see this theme of this NAD+ removing those hydrogen atoms throughout the process of respiration? The resulting The resulting carbon compound is combined with coenzyme A to form acetyl coenzyme A, known as acetyl CoA. Alrighty, in our next video, we are going to move on to the Krebs cycle. I am going to break respiration up into video 1 and 2 for you.